Hi everyone, my name is Rich Javier and I'm going to talk about the electron transport chain. So remember in Krebs cycle, glycolysis, and even in pyruvate oxidation, we generate NADH. So this NADH is going to lose its electrons and hydrogen in complex number 1, named NADH dehydrogenase. Dehydrogenase meaning it's an enzyme that will catalyze the removal of hydrogen in NADH. As a result, the electrons will remain in complex number 1. So our goal right now is to move those electrons through all of these complexes, but it's not that easy because remember the inner membrane is composed of phospholipid bilayers and its tails are nonpolar. So nonpolar tails and negatively charged electrons, they don't work together. So how do we move these electrons? It's through a carrier mobile called ubiquinone. So ubiquinone is also named as coenzyme Q. So this coenzyme Q will get the electrons in complex number 1 and take it all the way to complex number 3, cytochrome reductase. And complex number 3 needs to have another carrier mobile to move it to the other complex. Thus, the another carrier mobile is called cytochrome C. So as cytochrome C gets the electrons, it's going to reduce it. That's why the name of the complex is cytochrome reductase. Reduce it, a reduction is going to happen, meaning the gaining of electrons. And its job is to move it through the next complex, which is cytochrome oxidase. So in cytochrome oxidase, it's going to lose electrons, oxidation. So the electrons then will flow all the way down and attach itself to oxygen and some hydrogen ion to form H2O. Going back to the second complex is named succinate dehydrogenase. It's an enzyme, dehydrogenase, so it's an enzyme that will catalyze the removal of hydrogen in succinate and converting it to fumarate. That's why here, FADH2 is then converted to FAD. In the process, ubiquinone then will get its electrons from complex number E to carry it to complex number 3, another carrier through cytochrome C, then to cytochrome oxidase. So it's going to be a cycle, these electrons. And as the electrons passes through the ETC, through each of the complexes, it then pumps, it has this electric energy that pumps protons from the mitochondrial matrix all the way to the intermembrane space. So as the electrons pump through all of those complexes, it takes it, because it has energy, as it pops, it takes the protons up to the intermembrane space. So this intermembrane space will then have a large gradient, a large concentration gradient compared to the mitochondrial matrix. They'll be all up here. And ATP synthase then will use this large concentration gradient in its process. So protons then will then flow to ATP synthase. ATP synthase, that means it's going to generate in ATP. ATP synthase has two major parts, the FO0 and FO1. So these basically are two rotatory motors that's gonna help in the diffusion. Then in diffusion, it's going to mix ADP plus phosphate and turning it to our final product, ATP. So ADP plus phosphate equals ATP. The process is known as chemiomosis. So this part of the ETC is called the oxidative part because we are oxidizing it, like NADH being oxidized to NAD plus and FADH being oxidized to FAD2. This part of the ETC is called phosphorylation. Remember, phosphorylation is a transfer of any phosphate. So the transfer here is happening in ADP plus phosphate equals ATP, phosphorylation. So when you combine both of them, you have oxidative phosphorylation.